neurotransmitter has to be released from the axon terminal. But sometimes when your neuron releases the neurotransmitter, it could suck it back up. It's like it sends the signal and it says, nah, and it sucks the signal back. We call that reuptake. And the last way to get rid of your neurotransmitter is called enzymatic degradation. This is basically when an enzyme breaks down your neurotransmitter. And if you break it down, well, it's gone and you're not going to send the signal. So these are 3 ways to get rid of a neurotransmitter. One way, diffusion, have it leave or diffuse from the synapse. Reuptake, have the neuron that released it suck it back up into the axon terminal, reuptake. Or have an enzyme break down the neurotransmitter. That's enzymatic degradation. Those are 3 major ways your body will get rid of a neurotransmitter. But if you don't get rid of the neurotransmitter, what happens next? Once the cell that's receiving the signal gets that chemical messenger, what happens? Well, like always, it depends. There's options. It depends on what you want to do. Sometimes your neurons will tell another cell to do something. It will excite them. Or your neuron could tell a cell to stop doing something. It could inhibit it. If a neuron is responsible for stimulating a cell, telling a cell to do something, when that cell it sends the signal to, the neurotransmitter to, receives it, it's going to generate its own spark of electricity. And in order to do that, it has to do some of the similar steps of changing its plasma membrane, which involves depolarization. So if a cell gets told to do something, if it gets excited, it's going to generate what we call an excitatory postsynaptic potential, meaning it's going to generate its own spark of electricity. But you could tell a cell to stop doing something. If you tell a cell to stop doing something, well, it's not going to generate an action potential. How? You're going to cause it to hyperpolarize. It's going to become so negative that it cannot become positive on the inside. And it's like you've frozen that cell. It's going to generate what we call a inhibitory postsynaptic potential. It's going to hyperpolarize and stop doing something. Again, I'm just mentioning this for thoroughness. Like always, stick to the exam review. And when it comes to a chemical messenger, you actually have several different types. Your body doesn't just pick one chemical to send all the different types of signals. You have a different neurotransmitter for lots of different occasions. And some neurotransmitters are excitatory, meaning they will generate excitatory postsynaptic potentials in the recipient cell, meaning it will tell the recipient cell to do something. Or you have neurotransmitters that will generate an inhibitory postsynaptic potential in the recipient cell. It will tell the cell that receives it to stop doing something. And not only do you have different types of neurotransmitters, you even have different types of receptors for each neurotransmitter. And you don't need to know them. I just want you to know one particular neurotransmitter up close. It's called acetylcholine. Acetylcholine is a major neurotransmitter our body uses to help us to communicate with other cells, especially muscle cells, which is what we'll see in the next chapter. You spell acetylcholine ACETYLCHOLIN. Acetylcholine. These next slides are just going over different types of receptors for neurotransmitters. You could skip over those slides if you want. And again, we've already talked about the 3 ways in which you remove a neurotransmitter. Just one more time for completion. Diffusion is when the neurotransmitter diffuses or leaves the synapse. Enzymatic degradation is when an enzyme breaks down your neurotransmitter. And reuptake, also called uptake, is when the neuron that released the neurotransmitter sucks it back up. It takes it back. It reuptakes it. Okay. So now you know a lot about how neurons communicate. You know, how they're going to make their spark of electricity, where that electricity is going to go and how, things that could affect the speed, and what's going to be the end result. So now we could talk about some general things about neurons to kind of finish up now. For example, what if you get multiple signals? So one neuron, for example, could receive multiple signals from multiple neurons. Remember, neurons could communicate with each other in multiple places on that cell. So one neuron could potentially get a signal from multiple neurons that might be conflicting signals. So what do you do? What does a neuron do if it gets multiple mixed signals? Well, it has options like always. Something it's going to do to help come up with its own plan or analysis of information is called summation. This is basically how your neuron is going to listen to multiple signals at once to come up with a response. And like always, there are options. When it comes to summation, there are two major types. There's spatial summation and there's temporal. What's the difference? Again, they're both how a neuron is going to listen to multiple signals. But how? What's the difference? They're both called summation, meaning they're going to add some signals up. And the major difference is how you're going to add up the signals, how you're going to tally the votes, basically. In spatial summation, it kind of gives you a hint. It's called spatial for areas. This is where a neuron listens to all the signals it's going to get in one particular area. That's what you're seeing on this slide. This neuron in pink, labeled number 3, is getting a signal from 2 other neurons. And it's only listening to all the signals it's getting, basically at its dendrites. It's looking at all the signals it's going to get within a period of time in one particular area. That is spatial summation. It's adding all the signals from one area. That is spatial summation. And so every time it gets an excitatory signal in a particular area, it's going to generate the excitatory postsynaptic potential, depolarizing, aka becoming slightly positive, until it hits threshold and it will make its action potential to send the signal. But again, there are other ways to do it. Your body also has temporal summation. Again, just break the name down. You're still doing summation. You're still listening to multiple signals. But how now? It's called temporal because you're now listening to all the signals you get within a period of time. So this neuron can now listen to all the signals it's going to get anywhere, but only within a certain amount of time. That is temporal summation. And again, every time it gets an excitatory signal, it will generate an excitatory postsynaptic potential, depolarizing, aka getting slightly positive until it hits threshold, and it will finish off the whole thing, making its signal, making the action potential. So that's summation. So it doesn't matter if your neuron gets mixed signals. It could get excitatory and inhibitory signals. It'll just add them all up, and whoever has the most votes wins. That's all it is. And it's just whether or not you're doing it within a particular area, spatial summation, or within a particular time frame, temporal summation. And that's how your neuron will listen to multiple signals. And again, there are multiple neurotransmitters. There are multiple chemical messengers. Only one I want you to know is acetylcholine, the first one on this list. It's actually an excitatory neurotransmitter. So whenever a cell gets a signal from acetylcholine, it knows it's being told to do something. It's going to generate one of those excitatory postsynaptic potentials, depolarize, and if it keeps getting in enough signals, it would hit threshold and make an action potential and send a signal. And that's all I want you to know. There are other neurotransmitters. I can mention other ones, like on this slide. Some neurotransmitters send specific signals. Like this next one here called substance P. Substance P helps send pain signals. Kind of think the P in substance P is for pain. So again, when you're walking around barefoot and you stub your pinky toe on the table and life feels like it's over, you're feeling all that pain because your neurotransmitters are releasing substance P to let you know you're in pain. But again, stick to the exam review. There are some other more in-depth topics in here. Another one is called neural circuits. A neural circuit is just a group of neurons that are kind of gathering and providing specific types of information, but you do not need to know your different types of neural circuits, so you can skip over these. You do not need to know